Hi class, I want to quickly talk about mitosis. I'm just going to go through the very major steps of mitosis. Everything that we need to discuss in very small details, we'll do this in class. But here is just different phases and the major theme of each phase. Mitosis. So the mitosis, we need it for the cell division. So we're not going to discuss the cell cycle. So this is the last step of the cell cycle. So there are phases. One phase is called prophase, right? Another phase is called metaphase. Another phase is called Anaphase. And telophase, followed by Cytokinesis. So within the prophase, there's a breakdown of the nuclear envelope. And when the nuclear envelope breaks down, then chromatids, they condense and we can see them as chromosomes. We can see them in the shape X. So that's preparation for separation, for cell division. So that's the prophase, breakdown of nuclear envelope, and chromatids condense, and now we see them as chromosomes like that. In metaphase, all these chromosomes, and this is the copies, right? So that they line up on the imaginary plate, right? So there's 23 from mother, 23 from father. So that's the metaphase. That's when they line up in preparation of separation, being pulled apart to their respective poles. In anaphase, that's what actually happens. So one would be pulled to one side, another copy would be pulled to another side, like that, right? Like this. So they would be pulled to their respective sides. That's the anaphase. And do not forget, there's a 23 chromosomes on this side from mother and 23 chromosomes from father, so the copies of the original. And the same thing is here. And in telophase or the telophase, right? So once we pull them apart, so now we're making two new daughter cells. Because in the mitosis, at the beginning, we started with the original, let's say, mother cell, and was preparing through the cell cycle, through the gaps. It was multiplying on the material. So we're doubling in the material, so we can end up with two new daughter cells. So here, in the telophase or telophase, that's where the nuclear envelope develops or reappears again, because here we're going to have 23 chromosomes from mother and 23 chromosomes from father, right, this side. And here's one cell. And the same thing on another side, right, 23 from mother, 23 from father. And that will follow by cytokinesis because there's going to be separation. So, and we're going to end up with the two new identical daughter cells that has 46 and 46 chromosomes. It could be male, could be female, that we did not discuss. So I just wanted to quickly to talk about the mitosis phases, what happens with each phase. Obviously, there are a lot of other stuff that we will discuss personally in class, but I just wanted to quickly discuss the phases and the major theme of each phase. So if we want to make two new daughter cells, identical daughter cells, let's say there's a stimulus and we need to replace a new skin cell. We need to take an original and make a copy, right? So that's what happens. So you have a cell, and that cell has the original information, has 23 chromosomes from mother and 23 chromosomes from father, right? That make up 46 chromosomes. So that's what we inherited from our parents. So this 23 and 23 chromosomes, they never mix unless it is a sexual replication. But in normal cell replication, when you need to make a new system cell, they do not mix together, otherwise it would look different every time we get a new cell. For quick analysis, right, it's impossible to have all 23 chromosomes, but I'm just going to use like that, right? Let's say they represent 23 chromosomes from mother, that let's say would be mothers, let's say that would be father, right? Father, 1, 2, 3, let's say this 4 represents 23, and this 4 represents 23, 1, 2, 3, 4, right? And that all together, right, will equal to 46 chromosomes. So we never mix mothers and father, chromosome 1, right? Let's say this is the chromosome 1, let's say this is chromosome 2, let's say this is chromosome 3, let's say this is chromosome 4, right? So they do not mix unless it's a sexual replication. It is a sperm meets an egg. Egg has 23 chromosomes, and sperm has, that would be bad, and sperm has 23 chromosomes. So sperm fertilizes an egg, so it brings 23 chromosomes to an egg chromosome, plus 23, and you end up with 46. But they never mixed together, unless it is a sexual replication. So for the new skin cell, for example, we never mix them together. So now we have to make a copy for the cell division. That's what we discussed in mitosis. So we can have extra material to make new daughter cells.